So why do we want to get away from that? Because we've made such good progress. And one of the problems is that the patients that end up dying of CLL, almost half of them end up being associated with another cancer. So that the question is, does the chemotherapy cause the cancer, or do patients with CLL have a higher likelihood of getting other cancers? And that's partly true for both. There is a higher incidence of other cancers, of about a third more likely than average to get every subset of cancers compared to the average, except the acute leukemia transformation, which occurs in about 4 to 5% of patients that are treated with the FCR program, doesn't occur unless you have the chemotherapy part. And the Richter's transformation going into a large cell lymphoma has a very low frequency unless you have a chemotherapy program. And almost 3/4 of the patients that actually die in their first remission end up dying because of some complication. So that one of the things that we've tried to do now in patients over the age of 65 is to look at the what happens in the era of 2008 onwards and say, okay, if we can get away from chemotherapy, if we have a protocol available and we treat them with antibody regimens, as you see down the bottom, or lenalidomide, which is Revlimid regimens, or some of the new targeted therapies, compared to at the same time when we didn't have a treatment option for them, and we put them on an FCR-based regimen, does anything good happen? Well, the incidence of the solid tumors doesn't change. So I think that's just part of the association of the solid cancers along with CLL. But we do have a significant decrease in the Richter's transformation if we don't give them chemotherapy, and we have a halving of the likelihood of them getting an AML or myelodysplastic syndrome with time. So that we're beginning to try and identify the patients that are at risk of getting these complications so that we can increasingly get away from that. So after we analyze all this, what are the things that are problems? And Michael alluded to a number of these things, that the older patients don't tolerate the chemoimmunotherapy as well. That doesn't mean you can't do it, because there are groups in Israel and Australia that have modified the intensity and shown that if you just drop the dose down or extend the intervals in between, they can actually tolerate it quite well and have a very good outcome. But we do have these genetic changes that we haven't solved, the second cancers we haven't solved. But one of the things is that when you go into treatment of the CLL, you have a disturbed immune function anyway. Almost every element of the immune system is somewhat decreased. And that doesn't mean that you're a walking time bomb that's going to get some dreadful infection, because God has been good to us. He's built a lot of redundancy into the systems. So even though it's down low in some areas, the risk of getting a particular infection is actually pretty low. But one of the key things that we have to do now, if we're getting rid of the leukemic clone or the family of CLL cells, how do we go about rebuilding the immune system? And that's something that is being put in place now. And we'll probably be able to have a fairly effective way of markedly improving the immune system within the next 18 months or so. So we have a number of different ways that we can do it. We can do these, the B-cell receptor antagonists, the ibrutinib and idelalisib. The IMiDs, immune modulating drugs like lenalidomide, and there's another one that's coming along that appears to be 10 times as effective in stimulating the immune system. We can develop some things that are looked at in the lab to develop more rational combinations, and Dr. Plunkett and Dr. Gandhi in our institution are doing a lot of this. But before we were postulating that we were potentially curing some patients with chemoimmunotherapy, the only thing that was curing CLL was a stem cell transplant. And it was curing them because we put in a new immune system. And the new immune system killed off the leukemic cells because it would recognize them as foreign. So the thought now is, can we teach the patient's own immune system to do what a foreign immune system can do? And this, I think, is going to be the commitment that will occur in the next 18 months to 2 years in the United States. And as I more rapidly approach the age of 75 years, we don't have anything that's terribly damn good for that. So it just means I have to stick around and work really hard. Otherwise, I'll just be abandoned, stuck in a corner in a rocking chair and drinking red wine and reading mystery novels. Lenalidomide has been largely ignored, and it's partly because the company hasn't been very interested in it in CLL. But I find it a fascinating drug. It's very effective in myelodysplastic syndrome, in multiple myeloma. And if you start off with a very low dose, and in fact, in Frontline therapy, one of the first publications on this came from Toronto. But if you start at a very low dose, you can actually get a very good response. It's a medication by mouth. And one of the things that's of interest is, even though that in the chemoimmunotherapy, the patients with an unmutated immunoglobulin gene have an inferior survival, there's no impact of the mutation status of the immunoglobulin gene and survival of the patients that are being treated with these immune modulating drugs. And it was mentioned this morning, I think, by Graham or Dr. Johnson, the improvement in the immunoglobulin levels, which is a feature of CLL. The normal B cells are not making gamma globulin, but as you can see, the IgM and the IgG actually go up over time. So that's an element that we can do. One of the discussions this morning was, do we have a good response to vaccines? Well, we just put in place a protocol where patients that have finished therapy, we're going to be vaccinating them together with lenalidomide to see if we can improve the antibody response to the flu vaccine, the pneumonia vaccine, etc. So if we start off and look at the patients that didn't get the FCR base is in the green, so that if we start off with these alternative therapies, if anything, there's an improvement in outcome rather than a negative impact in outcome. So I think that rather than getting away from the one-size-fits-all, I think we have to figure out what the lesions are, what the genetics are, and see if we could approach it specifically. So over time, what's happened is that we've been getting away from starting patients off on the chemoimmunotherapy, so that the patients from 2004 on, the number of patients is in the blue, and the percentage of patients that were starting off on an FCR base has been continually going down. One of the problems, however, that we have is that the new agents are under the control of the pharmaceutical companies. They're very, very expensive drugs. The insurance carriers don't like you doing investigational studies with combinations of expensive drugs, so that we don't have the flexibility that we would like to have to get to the point where we don't need to start anyone off on chemoimmunotherapy. And this is something in North America, both above and below Niagara Falls, that we need to work on our governments to start saying, look, at least have a stash of drugs so that investigators can do things that they think are smarter than the insurance carriers think. We've been getting some background information, so that we've looked at patients that are 10-year survivors of CLL in our patient population, and the group in Winnipeg have their 100 or 200 patient studies. We have 470 patients, and we've looked at the patients that had cancers before they were diagnosed as CLL. And if you have some other cancer, you're more likely to develop CLL than if you don't have a cancer. But there are only certain ones. So we're going to set up a clinic within a clinic so that everyone that comes back to the MD Anderson every 6 to 12 months will have a full evaluation of whether they've had the required cancer screening test. We'll look for their skin cancers, we'll look for melanomas, and see if we can prevent these things from happening.